Welcome. We're very excited to have you here. We had a time to chat last night, and certainly we're excited. You're a pioneer and a visionary, and your work on the human genome has really established so much of what we look at today. But can you tell us a little bit about kind of where you started in science, where you are now, and where you think it's going in terms of your personal work?
Well, all my work in the human genome led to lots of arrows in the back, as Frank was referring to. But I started working on neurotransmitter receptors. My introduction to science was just learning how to ask questions. I was an adrenaline junkie, so I studied how adrenaline worked on the cells. Turns out in the early 70s, nobody had any idea. And I published my first paper as an undergraduate, proving that adrenaline worked on the outside of cells and that there had to be receptors there. Spent the next 10 years trying to isolate them, and moved to NIH in 1984, was given a large budget and freedom to do whatever I wanted. And I tried to get a grant to clone neurotransmitter receptors, but I was told that I had not trained with a molecular biologist, so I couldn't possibly do it. So with my new freedom at NIH, I retrained myself and my entire lab how to do molecular biology. In 1985 is when the first discussion about the Human Genome Project came up. And I thought, gee, I spent 10 years trying to get, one receptor protein from the brain. The Genome Project offered over 15 years to get every single protein. So that seemed much more viable approach. I just got excited about it. And because of the resources I had at NIH, I got the first automated DNA sequencer and published the first paper on sequencing 2 receptor genes with it. And then discovered a method for rapid discovery of human genes called express sequence tags. And that's when science discovery switched into politics and even more arrows in the back, because instead of being celebrated for coming up with changing discovery by 2 or 3 orders of magnitude, I got all these angry letters from Harvard professors and others that my paper wiped out 2,000 PhD theses.
And no one likes that.
And nobody likes that. So because they were spending 5 years on trying to do something that now the thing they were after was just one line in the paper from all the things we discovered. So the key to the advances was mathematics. We had so many sequences, we had to find a way to assemble them together. And that led to the notion of trying to sequence an entire genome using these new mathematical tools. So I met Ham Smith in the late 80s. He suggested he teamed up with me. He got the Nobel Prize for discovering restriction enzymes in 1972. In 1995, we ended up using this mathematical approach to sequence the first genome in history. And then it was logical to me that we would do the human genome that way. But nobody thought it was possible other than myself. I was fortunate. A company offered me $300 million to start a company to sequence the human genome using my method. It's a company that made sequencing instruments, and they thought if it worked, they would sell a lot of instruments, which they did. But we sequenced the human genome in 8 months instead of 15 years, which changed the course of things. That was now 24 years ago. It was supposed to be the start of a major revolution. That revolution has yet to happen, in part because the government just funded sequencing more and more genomes, not understanding them. So there's been now over 1 million human genome sequenced worldwide, but they're all haploid genomes, just with these short read sequences. They pile them on my genome as a reference or another reference genome. So everybody can find out how different they are from me, but it doesn't really tell them what they have. And so from my genome, I learned that I was heterozygous for ApoE4 alleles, which are, as you know, associated with increased risk for Alzheimer's disease. So I asked my colleagues at UC San Diego if I could get a brain MRI and an amyloid PET scan. And they said, no, you don't have any symptoms. It's unethical for us to do tests on asymptomatic people. Because they were friends, I convinced them to do it under a pseudonym. And they found that I had no amyloid. They said I had a brain of a 40-year-old. My wife said, no, he has a brain of an 18-year-old. But there was no correlation between what my genome was saying and what was found in my actual phenotype. And that's where I started asking different questions. The same is true for BRCA1 and BRCA2. Physicians have been giving the wrong information to women for basically since those genes were discovered. BRCA1 and BRCA2 give a woman a 50% lifetime risk of getting breast or ovarian cancer unless they have a family history. If every woman in your family has breast or ovarian cancer, then your risk goes into the high 90% range. So Angelina Jolie was a good example of that and had a prophylactic double mastectomy. A lot of women are trying to have prophylactic mastectomies based on just having the BRCA1 and BRCA2 test, which is totally inappropriate because the chances of them actually getting cancer are low. But shows those genes are not related to the cause of cancer. There's some distant relative to it. So we sort of found out how much we needed to know. And so I decided we had to start what should have started in the 1990s of doing phenotype analysis along with the genome. And without phenotype and clinical data, we weren't going to understand the human genetic code.
You know, it's so much of what we're taught in medical school is about being symptomatic without ordering extra tests. And now we are finally, I believe, starting to look at prevention differently. So where is that concept of prevention, longevity taking you now in your work and how you think about it? Because certainly, whether it's ApoE4 or other risks of cancer, breast cancer, even now today, women can't get an MRI through their insurance or ultrasound. So genotype, phenotype, sequencing, how should we be thinking about prevention?
So I started a clinical study to just do phenotyping on people who thought they were totally healthy and comparing it to the genome to start to annotate the genome properly. But the experiment went awry from the beginning. Our very first patient was a physician, and we found a major tumor under his breastbone that he didn't know he had. And we've gone on now with 10,000 visits. 53% of people who think they're totally healthy, we find some major disease. So people have thought of this before. So in the 1970s, when CT scanning came out, storefront clinics were opened all over the place, and people got whole body CT scans. But CT is just X-ray, so it's no different than a mammogram. It can't distinguish between a tumor and a cyst. So the breakthrough in MRI imaging came from my friend Anders Dale, developed new algorithms to post-process the MRI data. Some of you may know it's called restriction spectrum imaging. And it turns out tumor cells have slightly larger nuclei than normal cells. They have more water in it, but the water is less mobile, I think, because it interacts with all the DNA and the nuclei. And so it gives off a totally different MRI signal. Literally, tumors light up like light bulbs on the MRI imaging processing. So a 12-year-old could read these MRI images and diagnose cancer. It works best with solid tumors, prostate, et cetera. It doesn't work as well yet with breast or ovarian cancer. Ovarian cancer is the most complicated one because of ovarian cysts, follicles, et cetera. So we're starting a new version of what we did at Human Longevity, which was a for-profit company. It still is. But the trouble is, most of the data in genome databases are white European males, and very few females, very few people representing the rest of the diversity. because HLI required people to pay up to $20,000 for a visit. It's sort of become very self-selective of people interested in their health and having the means to do it. So it's a 1% clinic, and so it gives us very interesting data on the 1%ers, but not on the rest of the population. So we're starting a whole new clinic centered around women's health in the not-for-profit Venture Institute, so that we can bill insurance, we can get people from the diversity that represents the country. Women are reluctant to go through HLI. They will send their husbands, their sons, their boyfriends, sometimes all at once. But the number one thing we hear from women is they don't want to know.
Right.
We try to teach people that knowledge is power. You know, people want to control their health, and you do it like we heard from Frank and others. You try and control your diet, your exercise. You do things that you think you control it. Tumors don't just miraculously appear overnight. People can have them for 20 or 30 years. Medicine is based on symptomology. So until you're basically got stage 3 cancer, you're not aware that you have cancer.
Yeah, we have to change that paradigm because we talk about prevention, everybody thinks we take it so seriously, but we're actually really not approaching it the right way. So we have to think about it differently.
And all the work with the CT scanning created a new term called incidentalomas that every physician's familiar with. And they're taught in medical school that it's unethical to do tests on asymptomatic individuals.
We will change that. Talk a little bit more about diversity. We're talking about a lot of great innovations today, but how do we get these innovations out to diverse populations, equity of care, to our communities who are certainly not even accessing primary care? So how do we get them out into the clinical trials as well as part of this innovation?
Well, that's why we're doing it in the not-for-profit world, and we're seeking funding for this new clinic that will really specialize in trying to improve women diagnostic, because breast and ovarian cancer are still in the 1970s. So using what we heard about this morning, AI coupled with these new MRI algorithms, we're trying to make it so the primary thing is a breast MRI, not a mammogram, which is no different than what was done in the 70s. X-ray can't distinguish between cysts and tumors, as you know, and women have to go back and get ultrasound biopsies to see if something's there. So we have to do these studies in parallel to try and get rid of mammograms, but also because it's a not-for-profit world, there's various groups that want to sponsor disadvantaged groups to come in, and we're trying to make that a key aspect of it. Focusing on women is what's missing from the HLI experience, and teaching women that they can control their lives by getting this information early versus it's a death sentence or a horrible, you know, the next 20 of your lives are going to be spent in misery. So last night we talked about, Frank mentioned that we see the difference between a health span and the lifespan. Yep. So women live substantially longer than men, but they generally spend the last decade of their life in poor health. Yep. And so they have a longer lifespan, but they have a shorter health span. With this approach of getting so we can understand the genome, finding tumors early, so it takes chronic medicine and converts it to acute care. So when we diagnose tumors at stage 0, stage 1, there's easy treatments for them with surgery and radiology, virtually 100% cure rate. And so it changes the paradigm completely. But heart disease and cancer, as you know better than most, are the two biggest killers of women. Heart disease is catching up and maybe passing cancer. But with just pre-symptomatic testing, we could eliminate 2/3 of the deaths within 5 years of people dying from cancer.
Yeah, I mean, Kevin said it also as well. We know what kills people.
Yeah.
Whether it's cancer, heart disease, obesity, diabetes, neurodegenerative diseases. And if we know this, we have to approach it differently.
And this could change, by calculation, is $2 trillion to the U.S. government in healthcare costs. But as Kevin was saying, the whole system has to change. But I don't think it's going to be that difficult to change. Primary physicians can become healthcare coaches, guiding people through the system. Our biggest problem with the advanced data that we give people, they go back to their primary physician, don't have an effing clue what to do with it. They don't understand it. We had a billionaire that came in that we diagnosed. He had, with the RSI imaging, renal carcinoma. He went to Stanford to get a second opinion. They did a standard MRI, told him he probably had a cyst and not to worry about it. We convinced him to see somebody at UC San Francisco that actually understood our technology. They convinced him to get a biopsy. He did have renal carcinoma. So it's not just educating women and the public that they can control their lives. The healthcare system, the physicians are the ones that are most need of a new education system, and that learning to do early screens on people now is a totally different paradigm than people learned in medical school. The goal, we're adding all this data back to the genome to realize what we proposed 25 years ago, that if you know your genetic code early in life, and we understand more than the 1% we understand now, it'll be on top of the early diagnosis, we will be able to make predictions about everybody's life in a very precise fashion. So talking about neurons, it's known that your brain is totally genetically wired. It's also genetically wired to be plastic and adaptable. But in rodents, the same neuron shows up in the same exact location, plus or minus 1 or 2 microns, from animal to animal. The whole range of neurological conditions are pre-wired from the genetic code. We don't have the slightest clue of that yet because nobody's measuring these things. The government doesn't want to fund any personality test or anything to do with intelligence, with brain wiring, because that'll lead to segmentation and discrimination. So the goal is to keep living in the dark ages.
We have to break through.
We have the basis of something that could truly revolutionize medicine, but it's changing the payer system is going to be more difficult than probably changing anything else.
Yes, one would say. So continue with what you're excited about. We talked a lot in this room of what we're most excited about that's changing the course of prevention of disease, of how people live, of health span. What are you most excited about?
Well, this was a major revolution to me because I was testing my own equipment as I was building the clinic. And I had high PSA levels. I had a standard MRI imaging. I had 2 rounds of biopsies. And I was told by the best of medicine I did not have prostate cancer.
Yeah.
60 minutes in my own MRI using the advanced mathematical systems. My own radiologist had to come and tell me that I had 2 advanced tumors that were totally missed less than a month earlier
remarkable,
by the best of medicine.
Right.
And fortunately, it was just growing down the vas deferens, had not metastasized.
Yeah.
Leonardo da Vinci did my surgery, and I had to go back a long way. And
See, we're going back and going forward.
That's right. And I've been cancer-free for 6 years.
Well, that's wonderful.
Had I not stumbled in and been doing my own experiment, I would have died sometime in the last 6 years.
But you advocated so much for yourself to get yourself to that place, and we have to make that a standard of care for patients.
I'm an experimental scientist. I always do the experiment. If women are afraid to do the experiment, they're going to miss out on this revolution and continue to die from diseases that could have been totally eliminated because they're waiting for symptoms to appear instead of getting rid of them early on.
So, and it's wonderful to hear that part. And let's go to scientists for a moment. You're a scientist, you're a researcher for your entire career. What advice would you give to scientists and researchers starting out, embarking, mid-career? It's frustrating. They're long journeys. Kevin talks about 28 years of his journey. These are journeys. So is there advice or words of wisdom, inspiring thoughts?
The most important advice I got from Nate Kaplan, who died in the 1980s, one of the most famous biochemists. He discovered Cohen's MA with Fritz Lippmann. He said most scientists fail because they talk themselves out of doing the experiment. And it doesn't matter whether the experiment's something you're doing in the lab, the experiment is doing you're going to a new clinic to get screened even though you're sure you're perfectly healthy. Everybody who talks themselves out of doing an experiment fails. So number one advice, don't talk yourself out of doing the experiment. Be an experimental scientist, do the experiment. And the second thing is that, pick something you're really passionate about.
Always.
And that way, you do like Kevin, 25 years blows by because you're passionate about what you're doing. And I've had the most exciting life as a scientist that I never thought I had until I got drafted off my surfboard in 1965. I thought I was going to have a very different career. I didn't even know about science until I got dumped in the middle of the medical corps in Vietnam and had to learn about science and medicine. But that it was life transforming for me, and I've always taken the step and done the experiment. And I do that with every aspect of my life. I got my pilot's license at age 72 during COVID.
Wow.
And now fly myself cross country all the time. So
I love it.
It's never too late.
That's wonderful. Well, I hope you're still on your surfboard as we think about.
I did sell my surfboards this last year. I had knee surgery. I ran a mile, and I told my physician, she goes, "That's great. Don't do it again."
So it's prevention and being careful.
That's right.
All right. It's really been inspiring to hear your thoughts on your career, on the genome, where you were, where we're going. So thank you so much.
Thank
it's really
you.
Been
Yeah.